Welcome to Biochemistry Simplified. Today, we explore amino acid classes, the foundation of proteins, and life itself. We'll break down the different types, their structures, and why they are essential for biological functions. If you're ready to master biochemistry, stay tuned and don't forget
Welcome
to
to
like,
Biochemistry
subscribe, and
Simplified.
share.
Good day, everyone. My name is Dr. ELIC. I'm back to exciting lecture on amino acids, and we want to take it up from where we left off in the classification of amino acids. Okay, and in the last uh, class, we were able to look at the different classes of amino acids based on their functional group, their R group, and we did mention that we have 5 different classes on that basis, and um, we are able to look at the five. We did mention the 5 of them, which are the non-polar R group, aromatic um, arrow group, polar, non-polar arrow group, aromatic arrow group, positively charged arrow group, negatively charged arrow group, and the polar arrow groups. And we did look at each of these groups in some good details, and we were able to cover 4 of them, which are the non-polar R group, aromatic R group, positively charged and negatively charged R groups. So that's just like a quick recap of what we've done. Today, we'll be doing the last on the list, that's looking at their specific structure, how they are represented, and we are looking at the polar group, and we say that they are the uncharged but polar arrow groups. So that's where we take it up from today. And so we look at uncharged but polar arrow group. Okay, so we'll begin here, and the first on the list we want to look at today is the structure, is the amino acid serine, which is a polar arrow group, but that is uncharged. And we are looking at the structure of serine again. Like I encouraged you um, in the last class to always make sure you try to draw the structures continuously, and they say that repetition is a lot of deep and lasting impression. So as you continue to draw these structures out, you get pretty used to them. So we are looking at serine, and the structure of serine is COO minus, which is your carboxylic group attached to your alpha carbon, and this is attached to the amino group. So your serine here is your CH2OH. That is the R group, and that gives you serine as the R group. And this is serine, and the 3-letter um, nomenclature is CRE, and the single letter is S. So that's for serine. And we want to look at again threonine. So we'll look at threonine here. So we have COO minus, C, NH 3 plus. Then for serine we have C and OH attached here, and H attached here, and we have a methyl group attached right here. And so what we have here now is your serine. So this is your, sorry, actually threonine. This is threonine. Threonine, the three-letter word, sorry, three-letter word, and the single letter is T. So actually there's no dot around here. So I'll just, sorry about that. So that's your training. I want to look at a third one. Actually, we've treated 15 of the 20 common amino acid, and in this group, we'll be treating the remaining 5. I want to look at another amino acid right here, and that will be the amino acid cysteine. We'll talk a little more about cysteine after this, looking at these groups. So you have NH3 plus, and you have CH2 SH. Okay, and this group right here is what we refer to as cysteine. Cysteine. Alright, the three-letter word here is C, and the single letter is C. Okay, so we have serine, cysteine, and threonine. For want of space, we'll be looking at the other two, which is asparagine and glutamine, in the next page. Okay, so coming right here, we've looked at the serine, the threonine, and the cysteine. Now we want to quickly look at the asparagine. Asparagine is your COO minus attached to your alpha carbon. By now you should be pretty used to drawing out these structures. So asparagine is actually from the negatively charged amino acids that we look at, which is aspartate. So right here, if it's the negatively charged, you have the carboxylic group attached. But for asparagine, the negatively O minus here is actually replaced with an amino group right here, and that gives you is actually referred to as the amide of aspartate. So the whole of this is actually what gives you the amide of aspartate this here, and is referred to as the amide of aspartate. Good. And the whole of this is actually your arrow group. The whole of this. Okay? So the whole of this is your arrow group, and um, that forms the asparagine. And asparagine is written as, the three-letter word is ASN, and the single letter is N. So the next we want to look at is still on the uncharged but polar group, is the glutamine. So your guess is as good as mine, just as you have for aspartate in the negatively charged amino acid. Remember that, recall that we also had for glutamate. So just like we saw here now, having the amide of aspartate in doing the glutamine, we begin to see the amide of glutamate. So let's quickly look at how the structure looks like. So you have your carboxylic group attached to your alpha carbon. This is your H, and this is your NH3 plus. Then you have for your glutamate CH2, which for your glutamate should be attached to a carboxylic group right here, double bond O, and O minus. But for the this group, it is now attached to an amino acid, and that forms your glutamine. The whole of this here is referred to as your amide of glutamate. Amide of glutamate. And the whole of this, from those points down this way, forms your R group, alright, of the amino acid, and that is what gives it the specific amino acid glutamine. And the three-letter word is GLN, and the single letter is Q. Great, so that is all for the classification. So we've successfully classified the 20 common amino acid based on their R group, their reactive R group that gives it a specific amino acid. Now we're going to be looking at some things we need to know about, some additional information about some amino acids, and we'll be taking that in the next. Right, welcome. We are looking at some additional information on some amino acids, and in the just concluded amino acid classification group, which is the polar but non reactive arrow group, that uncharged but polar arrow groups that we looked at, we saw the amino acid cysteine. And I did mention I'll give you a little more information about cysteine. So we'll be looking at additional information on cysteine. So cysteine easily dimerizes to form something called cysteine. So cysteine easily dimerizes. So by dimerized, we mean it implies when two molecules come together to form a single molecule. So among the 20 common amino acids, cysteine have the ability to easily dimerize to form a new molecule called cysteine. Okay, so to form something called cysteine or cysteine. Again, the pronunciation is not as important as the chemistry. So that is cysteine. So we'll quickly look at a reaction on how cysteine can be dimerized to cysteine. And with that, we look at the structure of cysteine again. So this is cysteine, C, alpha carbon, NH3, NH3, and we have CH2SH. All right. So this is a molecule of cysteine, and we said that to dimerize is when two molecules of a particular compound form to come together to form another single molecule. So this is cysteine now. So we're gonna look at joining another structure of cysteine together to show how the dimerization takes place. NH3 plus. Okay, so let me adjust this a little bit. All right, so we have another CH2SH, and this is another molecule of cysteine. Good. So the cysteine undergoes a reaction. Is actually an equilibrium reaction, a forward and a reverse reaction. And in the forward reaction, we see um about that, we see 2 hydrogen plus 2 electrons giving off. Okay, and in the reversed reaction, we see in the reverse reaction, we see 2 hydrogen plus 2 electrons coming in. All right. So the 2 hydrogen that is actually given off during this dimerization reaction is actually the hydrogen that is attached to the S. So these 2 hydrogen comes together and they are given off in the process. Okay, so these 2 hydrogen atoms are given off, and what you have produced is your cysteine. So you have COO minus CHNH3 plus, then you have your CH2, and instead of having your SH, you have S. Okay, and that is for one of the molecule, one H is given up, and you have this second molecule attached just right to the second S here. So what you have is that when this OH, when this hydrogen atom, 2 hydrogen atom leaves or is removed, okay, then you have this S, this S and this S joining together. Okay, so let me do that in another color so that you see it clearly. So let me do it in blue. So you have the S, this S and this S over here. All right, so that S and that S joins together to form a disulfide bond. Okay, so I'll go back to my red to complete my structure. Okay, so you come here now and you see that this S, which is this S of this other one, is attached right to the second cysteine molecule. So you have CH2, then you have your alpha carbon down here, which is bonded to an H and an NH3 plus. Then you have your uh, COO minus. All right. So um, this structure, so from this whole structure, I just want to have COO minus CH and H3 CH2, and you have your S. One of the H has been removed, and this is merged to another S. Okay, CH2, and you have attached to uh, your alpha carbon, and this is attached to an H, and this is attached to an NH3 plus, and this is attached to a carboxylic group. So you see this bond right here. This is actually where your dimerization has taken place, and this bond here is referred to as your disulfide bond. Disulfide bond. This is referred to as your disulfide bond. That bond that connects these two S sulfide after the O, after the H, the hydrogen atoms have been taken off as seen in this reaction. So the whole of this, if I do that in blue, the whole of this is one molecule of cysteine, and the whole of this is another molecule of cysteine. All right, so that's how the two molecules are formed. Thus, 2-cysteine amino acid can actually oxidize to produce 2-cysteine dimers. As you can see, a dimerization reaction has taken place right here, and this is one molecule of cysteine and another molecule of cysteine that have dimerized to form by the giving off two molecules of hydrogen and two molecules of electrons to form your cysteine dimers, two cysteine dimers. Okay, now in the same vein, a cysteine dimer, which is this two, right? This is a cysteine dimer. A cysteine dimer can again be reduced, and to be reduced is the addition of the two molecules of hydrogen and two molecules of electrons, that two electrons rather, that was given off. When you bring it back in, it actually produces two molecules of cysteine. That's why you can see it's a forward and a reverse reaction. So it is a reversible reaction. So in the next page, Okay, so in this page, we just quickly want to note certain things. So we say note from the dimerization reaction that we saw now. We saw that two molecules of cysteine, two molecules of cysteine amino acids can actually oxidizes to produce two molecules sorry about that. Oops. To produce two molecules of cysteine dimers. Okay, that's first point to note. Other point to note is a cysteine molecule, a cysteine dimer reduces, so the addition of hydrogen is a reduction, while the removal of hydrogen is oxidation. So it reduces to produce two cysteine amino acids. Okay, that's pretty much it. And another thing to note is that in the forward reaction, of the dimerization reaction we saw now, in the forward reaction, all right, two hydrogen plus two electrons are lost, worthy of note, and that implies oxidation. Okay, why in the reverse reaction, lost, worthy of note, and that implies oxidation. Okay, why in the reverse reaction, two hydrogen plus two electrons are gained, and that is what? Reduction. Very important to note. So that is dimerization reaction. So another additional information we want to look at is with the tryptophan molecule. So I do that in the next page. Okay, another additional information that we'll be looking at is tryptophan, not just tryptophan and thyroxine. Tryptophan and tyrosine and to some extent, and to some extent, and to some extent, we have phenylalanine. So, sorry, we have phenylalanine, not tryptophan again. So, and to some extent, phenylalanine. It's pretty interesting. My mouth is saying something else and my hand is writing something else. So, tryptophan and thyroxine, and to some extent, phenylalanine absorbs ultraviolet light. You observe that tryptophan, thyroxine, and even the phenylalanine are all examples or structures, amino acids under the aromatic amino acids with the aromatic R group. So they absorb these three amino acids. They absorb ultraviolet light. Ultraviolet light, UV light, strongly. Please take note. They absorb UV light strongly because they have the aromatic group. Very important to know. Because they have the aromatic group as the aromatic R group. Aromatic groups. So they have very strong absorption at about wavelength of they have very strong absorption. They have very strong absorption at about wavelengths equal to 280 nanometer. This explains why protein absorbed UV light. It is because of the tryptophan, the thyroxine, and the phenylalanine that is present in proteins. This gives us an analytical tool by which we can measure or identify proteins. Very important to note. So it's not just these ones, there are some other amino acids that like I said, that are not so common. These amino acids are not so common, but they are derived from these common amino acids, and they are modified after the proteins have been synthesized. Okay, so you note that they want to look at some uncommon, as it were, amino acids that are derived from some common amino acids after they have been modified and synthesized. What are we saying? We are saying that, in other words, they are also uncommon amino acids that you will find in proteins, but they are incorporated while the proteins is being built one amino acid at a time, strung together as beads and strings. These common amino acids or proteins are built using the 20 common amino acids. Then after that, there's a modification. The modification implies either there is an addition to or subtraction from a particular amino acid residues. So we want to quickly look at these uncommon amino acids. So we'll come here and do uncommon uncommon amino acids. Alright, so uncommon amino acids, we want to look at some examples of this uncommon amino acid, and one of them is the 5-hydroxylysine. 5-hydroxylysine. So we want to draw out the structure of lysine quickly. So we have your C, which is your alpha carbon, bonded to your H, and your amino group, NH3 plus, and this is bonded to your carboxylic group, O minus, alright, and that is bonded to CH2, CH2. Remember, lysine, we have 4 methyl group attached to it. So CH2, CH2, 4 of them, and this is attached to an NH3 plus. So you remember that the amino acid lysine is in the positively charged amino group. So this is lysine, but at the fifth position from the carbon, you have an hydroxy group attached. So we have carbon 1, which is the alpha carbon, 2, 3, 4. Okay, fourth position, and that's at this point you have your OH attached. Okay, so what you have in naming this now, you have your 5-hydroxy. It's named as your 5-hydroxy lysine. Okay, it can also be named, remember that this carbon right here is referred to as the alpha carbon, and of course this carbon is the beta, and this carbon is what? The gamma, and this carbon right here is the delta. Alright, so you can name this also as, or can be named as your gamma hydroxy lysine. So instead of 5-hydroxy lysine, 5-hydroxy lysine is counting from the number of carbons, and that is 1, 2, 3, 4, 5. And what you have here is your gamma hydroxy lysine. That's pretty much it. You have both your 5-hydroxy lysine or your gamma hydroxy lysine. And important to note is that this particular amino acid is found in collagen. It is found in collagen, and collagen is a connective tissue protein. So we'll look at another one quickly in the next page. Okay, so the next one we want to look at is your glutamate. So in this case we'll be looking at your gamma carbon glutamate, or that could also be named as your 4-carboxyl glutamate. So drawing the structure of glutamate, you have your alpha carbon that is bonded to your H. See, sorry, let me write this out completely. You see double bond OO minus. This is bonded properly here. See, and of course you have your NH3 plus attached here in your alpha carbon, and you have for glutamate you have CH2 attached, CH2 attached, and now instead of your CH2 here, a carboxy group. So we remove the CH2 there. A carboxy group is now attached at this point. So you have COO and O minus. Okay, and of course your normal glutamate will have C double bond O minus. You get that? So this is you have 3 carboxylic group attached here, but this is your normal carboxylic group in your amino acid structure, and this shows your carboxylic group with the negative charge that groups glutamate into a negatively charged amino acid. So what we have here is your alpha carbon, here you have your beta carbon, and of course here you have your gamma. So what we have here is alpha carbon glutamate. Alpha, I think this should be alpha carboxyl glutamate. Great. So that's basically for the additional information we want to be given during this lecture. In our next set of lectures, we'll begin looking at peptides and proteins.